Hello, everyone. So good afternoon. Today I have the pleasure of introducing Chris Voigt, who's joining us from across the bay from UCSF Bioengineering. So Chris did his bachelor's degree in chemical engineering at the University of Michigan. Went on to do his PhD at Caltech. He had 3 advisors there, I believe. Francis Arnold, Zhengang Wang, and Stephen Mayo. And so he worked on using computers basically to improve directed evolution of proteins, which is something that I know many of us work on. And so he then came here to our own UC Berkeley to work with Adam Arkin in engineering bacillocellulose and some genetic circuits there. He's since been at UCSF since 2003, where he received an NSF Career Award. He's currently a Packard Fellow and is really viewed as, I think, a leader in synthetic biology. So in addition to being have a great research record, he's also an excellent mentor and recently landed a couple of his alumni at what might be one of the more prestigious universities on the planet. That's UC Berkeley. So I'm talking about John Christopher Anderson and Danielle Tolman-Ursek. So we're very excited to have you here today, Chris, and so anxious to hear about your work on programming cells by refactoring prokaryotic gene clusters.
Oh no. Danielle ran and she just keep going. Yeah. I'm such a good mentor. None of them show up to my talk. Yeah. It's always great to be back. It's great to see so many familiar faces in the room. I, of course, was a postdoc in the Arkin lab back when he was in this building, and this was the room that we used to give group meetings in. So it's nice to be back in a little bit of a different context. So today I'm going to actually talk about one of the applications that we've been working on in the lab, which is to take a lot of the foundational work that we've been doing in synthetic biology to really create a basis to program cells. And I'll show how we're applying this to one problem where we're trying to go into very complex functions in bacteria and rebuild them from the ground up. And the particular system I'm going to talk about is for nitrogen fixation, which I think is peripherally relevant to some of the things that are going on at EBI. So this is usually the slide that I start out with, and it is something that really inspires our lab. And it's showing the growth of several technologies over time, where in the blue line, that's what's known as Moore's law, where the number of transistors per chip has been doubling about every 2 years, and this has led to electronics and computers and so on. 2 technologies in biology have been growing even faster than that. One is DNA sequencing, which has been growing very rapidly, and then trailing that has been DNA synthesis. And so this is the ability for me to be a monkey on the keyboard, type out whatever DNA sequence that I want, send that off in an email to our dear friends down at DNA 2.0, and about a week later they send back the DNA. And so this gives us a design capacity that wasn't there previously. It certainly wasn't there when I was a postdoc or certainly graduate student. And a lot of what we're doing in the lab is trying to figure out how we can harness both the capacity as well as our ability to design systems based on this technology. So really what this is doing from a very high level is it's reversing the tide of bioinformatics, where over the last 20 years, sequence databases have been populated by all of the sequencing efforts that have been going on, from individual organisms to metagenomics, where entire populations are being sequenced. And this has led to, the last time I looked it up, there are now 108 million sequences in NCBI from 260,000 organisms. And so that's a huge amount of DNA information, most of which we don't know what it does. We might have a guess, but there's very small fraction of that has actually been annotated with any accuracy. So that's DNA sequencing. DNA synthesis now allows that loop to be completed, where you can take that information out of those databases, and I can send that information either directly what was in there or in some modified form, codon optimization and so on, off to a synthesis company where they print out the DNA using robotics. You can figure out what all those sequences do and so on. And so there's a real capability to go into those databases and start screening for interesting functions. Just to give you one example of something that we did and published about a year ago, we were interested in pathways that would make methyl halides. They're a precursor for a number of different chemicals. They're also used as part of silicone processing. In the chemical industry, it's natural gas and hydrochloric acid. We wanted to make it from sugar and salt, so we started looking for genes that were able to do this. And so instead of going out into the natural world and trying to find an organism that had this function, we had a scaffold of a protein that we thought might be able to do this. We then used that to search the databases for all possible sequences that might have that function. Sent that off to synthesis, printed out about 100 of them, and then screened them all for function. And so this is a single gene. Those that had the highest activity came from sort of some unusual places like ice plant, which grows over by the ocean, bok choy, rice, bacteria, uncharacterized organisms, and so on. And so what the synthesis allowed us to do is, without touching any of these organisms, without touching their DNA, we could print out all the possibilities, screen them for function, and so on. And so this is for a single gene with a simple function. A lot of what we're doing now is trying to do a very similar thing, but to go after much more complex multi-gene functions. And so this is where we've started to get interested in gene clusters in bacteria. So often a very complex function is encoded together in the genome. This facilitates genome transfer and evolution and so on. And so you have a unit of DNA in the genome, and inside that unit you have everything that's necessary and sufficient for that function. So for example, nitrogen fixation, which I'll be talking a lot about today. This is a 25,000 base pair gene cluster out of Klebsiella. It has all of the necessary genes for the nitrogenase, for the pathways that make the cofactors for the nitrogenase, the electron transport, and so on. And these gene clusters just have an enormous range of functions. We also work with type 3 secretion, which is a hypodermic needle that pumps out proteins out of the cell. Many cellulosomes are encoded within gene clusters. Things like light harvesting complexes and photosynthesis, and there's a whole plethora of clusters that make a variety of molecules that are relevant for chemicals and pharmaceuticals and so on. The problem is that these are often extremely complex systems, and very little is known about their regulation. And it's critical that all of the components be expressed at just the right levels in order to get the function that you're interested in. And so what we've been trying to do is to go in and using all of the tools that we've been developing from synthetic biology, plus DNA synthesis, rebuild these systems in a much simplified form from the ground up. And so what this allows us to do is to wipe out all of the native regulation, all of the unknown unknowns in the system. You want to get complete control over the function of that system. You want to turn it on at one point, turn it off at another point, control flux, optimize the system, and so on, and not have the organism fighting back with you. And then this is sort of a intermediate step between where we are now and whole genome engineering, which I think is the future in a decade or so, and I'll come back to that. So this is an example of a regulatory network that controls a gene cluster that we've been very interested in that I'm actually not going to talk much about today. This is the 34,000 base pair cluster that controls type 3 secretion in Salmonella. It's a large nano machine that pumps proteins from inside the cell to the exterior. It's very useful if you want to purify a protein, if you want to get something like a cellulase outside of the cell because the substrate doesn't diffuse in. So we were very interested in the cluster. We started going in and trying to engineer it, and there's a whole slew of problems when you start to do this. This is an example of the regulation, both on how this cluster is regulated and how it fits into the global regulation in the cell. And Salmonella does very tight control over when it turns it on and when it turns it off, because as it's part of one stage of an infection, if it turns it on at the wrong time or to the wrong amount, that's one mechanism that the immune system finds the bacteria. And actually it got so bad that this goes back to when I was in the lab in the early days. And I had a postdoc, and he could not clone one promoter out of this 34 kb. So this is a 100 base pair region. He couldn't clone it to make a GFP reporter, and I said, I'll do it. I went in, and you guys don't know me that well, but that's a disaster waiting to happen. And I couldn't do it. And then just this year, what was encoded within that promoter was a small RNA that interacts with the porin system in the cells. And so when you put that at high copy, it makes this small RNA, it goes and affects porins and so on. And that's a classic example of where you get tripped up by unknowns in your system. And so rather than constantly trying to use bubble gum and sticks to fix all of these leaks as they emerge, we started to think what we really have to do is go in and just wipe out everything we don't know, only use parts that have been well characterized that we trust, and rebuild it from the ground up. And even if we start out with a system that's less efficient but fully understood, that's going to be a better platform in the long run for optimization. And so this is a concept that was taken out of computer science refactoring, where the idea is that you're trying to create something that has the same function but is rewritten. So if my Windows Millennium keeps crashing, what you can do is refactor the code. That means you rewrite the code so you still have Windows, it's still the same program, but you've rewritten it to be more stable. And so if you look at the details of the code, it's different, but at the high level, it's supposed to be doing the same thing. In terms of refactoring a gene cluster, the idea is to start out with the sequence information of that cluster, go in, take out all the genes that are not essential for that function and for the particular use of that function, remove all the native regulation. That's where a lot of the problems start to come up. So all the transcription factors, everything that you know that is just regulating the genes. Remove all the non-coding DNA, and then we go into each of the open reading frames and randomize the codons. We literally say go as far away from wild type as possible. We're not trying to optimize expression, we're just trying to wipe out all of the internal promoters, the internal terminators, ribosome binding sites, get rid of everything except for the fact that it's coding for a particular protein. And this results in a set of genes that have about 30 to 50% codon identity, and they've just been randomized to get rid of all the unknowns. And we're running at the same time that we do this all sorts of programs that we've written and others have written to search it for unknown sequences that might be functional. We then reorganize those genes into operons, and the organization follows what makes sense to us versus the way that it was laid out in nature. And then we use synthetic parts, synthetic ribosome binding sites, synthetic terminators, synthetic promoters, and so on, so that for every single individual piece in that system, we understand what that piece is doing. And then the last step is to build a synthetic controller. And this recapitulates all of the necessary sensing that the cell has to do to know that it's in the right environment or in the right energetic state in order to turn that system on, the circuitry that integrates those sensors, and then what we call wires, and I'll explain that later, which turn on the different aspects of the structural system. We usually carry these on 2 plasmids. Because this system shares no DNA identity with what we started with the wild type sequence, we have to use DNA synthesis in order to construct it, usually in pieces. Okay, so the outline for today is I'm going to start out by kind of going over a set of projects we've been doing in the lab that are around building that controller that's regulating when the system turns on, the synthetic sensors, the circuits, and so on. And what this is really an agglomeration of the different projects that we've been doing in the lab that have all been leading up to this point. And then we're going to talk about how we've been applying this to refactor the nitrogen fixation pathway out of Klebsiella. Okay, so first, the basic idea of the genetic controller is to have a set of sensors that are responding to environmental stress, that are sensing the cell state, sensing cellular stress as well. And then you have circuitry that's integrating that information, and then you're controlling different aspects of what's happening in the cell. And the analogy that I like to use is if you think of a chemical plant, my background's actually in chemical engineering, and you're designing that chemical plant, use programs like Aspen to go in and regulate the flow it's been a while, the flow of the chemical through the plant. And so that's a sort of a series of tools that you use in order to do that. And that's kind of the equivalent of metabolic engineering. There's a series of tools that tell you how to optimize a system for metabolic engineering. For the circuitry, it's going to be a different set of tools, and those tools don't yet exist, and I'm going to kind of describe what we've been doing to get there. And it's the equivalent to a control room in one of these chemical plants, where you might use Verilog or VLSI in order to build the circuitry that's then controlling the plant itself. These are a number of different systems that we're actually designing or building, and I'm not going to talk about all of them, just the one in the middle. And the basic idea that I want to get across is that if you have a series of sensors, it's often the same sensors that you need. You need to sense oxygen, temperature, different stresses, and so on. You then need to integrate those using circuitry, so you need platform circuits that can be configured in different ways. And then you need some way that you can take the output of the circuits and have that control different systems. And so for that part in the middle, if you have a good platform that can be reconfigured different ways, there are a lot of different applications that you can address. So our lab and others have been building a large library of sensors that can respond to different things like oxygen, methylation, stress, byproducts, and so on. And essentially, I'm not going to go into the details of that, but if you have the transfer function, which is how the sensor responds as a function of input, that gives you the information that you need in order to connect to that sensor then to a circuit that can start to integrate that information. So in the case of the oxygen sensor, it's the activity of that sensor in the on and off state. In the methylation sensor, this is as you dial in more inducer, the system turns on. And the shape of that curve allows you then to connect it into a more complex system. This is an example of that, which we've been doing over the years, and I think it's a good visual example of how you can start to do signal processing on top of circuitry. This is a light sensor that we built a number of years ago, where it would give E. coli the ability to respond to light. The graph in the middle is the transfer function of that sensor. So as you add light, it turns that sensor off. And then on the right, you see a plate where if you shine an image on that plate, it records that image. So a very simple signal processing thing you can do is take an inverter, which is just a repressor that then shuts off a output promoter. So if the input promoter's on, the output promoter's off, and vice versa. And so when you connect that to the sensor, then the output is the opposite. And so those cells that are in the light are dark, those cells in the dark are light, and so on. And we can actually get pretty complex with the types of signal processing that we do. Last year, we published an edge detection algorithm where we took a simple program where if every bacterium's a pixel and every bacterium's running this program, then they would be able to detect the edge. I'm not going to go into the details other than there's a series of circuits that we had to be able to put together in order to do this. And then now the bacteria are able to calculate where the edge is between the light and the dark regions. And so as we've been putting together these programs, both this program and a lot of the others that we've been working on in the lab, we started to get a little bit jaded with respect to how we were going about the design process. It often had this very kind of hokey feel where you're combining different types of biochemistry in order to kind of recapitulate. So you take this promoter, you put a transcription factor, you've got a phosphorelay, an inverter, and all these different things with very different chemistries and so on. And it started to feel a little bit like a Rube Goldberg machine where you have your inputs and then you have to go through this crazy series of steps between the inputs and the outputs in order to get the type of logic that you want. And it works very well. We can build systems that do it, but we're not developing the underlying pieces that can then be used in a lot of different ways. It's always a fix for that particular system, and then when we go to try to design a new one, it's a completely new set of biochemistry and so on. And so we've been trying to address this problem. So a good example of this is an AND gate that we built a number of years ago. This was Chris Anderson's project when he was in my lab. The details don't matter. Essentially, what it's looking at is the activity of two promoters where the output of the circuit is on only when both of those promoters are on. And the underlying biochemistry that allows you to perform this AND gate function is the interaction between a tRNA and a mutated polymerase that has a codon that reacts to that particular tRNA in order to produce the polymerase, which then turns on the output. It works very well. One of the things we demonstrated at the time is that it's very extensible because both of the inputs and the outputs are promoters. So I can take any promoter off the shelf, basically, and with minimal tuning, have it be an input into the system. The output is a promoter, and again, I can use that output to drive different processes. And so it's a very modular system. And it had a very strong response. It produced a large fold change when both of the promoters were active. The problems were that one thing is that it wasn't scalable. So this particular tRNA and this particular interaction allowed us to build one gate, and then we're done. And we can't use that design strategy to go out and punch out 100 different gates because that biochemistry was so unique to the circuit. Required a lot of DNA, and there was very, very slight toxicity, which you probably, less than 5% in terms of the change in the growth rate, which you don't notice when you're just building an AND gate. But when you start putting these larger systems together, as we've been doing, then those 5% really add up, and you end up with a program that can be quite toxic on the cells. So we went in and we decided to take a step backwards in the types of circuitry that we were designing, where we went for the very simplest type of biochemistry systems that really work well, that would be a good platform for scaling up. And to this end, we designed a transcriptional NOR gate. And this is a very simple extension from the NOT gate that I showed earlier, where what we have are two promoters in series. And those two promoters act like an OR gate. So if either promoter is on, you get transcription. It turns on a repressor, which then turns off an output promoter. So if either of the inputs are on, or both are on, the repressor is produced and the output is off. And so this is the NOR function. It's very simple. It works very well. It's very fast. And importantly, it can be layered in order to create more complex circuits because the inputs and the outputs are both promoters. The output promoter from one circuit can then become the input promoter to the next circuit. So you can layer these out like integrated circuits in an electronic system. And it's also very scalable. As many orthogonal repressors as you have, that gives you the number of NOR gates that are available to you. And so we went out and built this and demonstrated that the system works very well. If we take the transfer functions of two input promoters, so this is as the PBAD promoters turning on to ribinose, the PTEP promoters turning on to tetracycline. If you put those two promoters in series driving YFP, the system behaves like an OR gate, where if either inducer is present, the system turns on. If you invert that with a repressor, then it becomes a very digital NOR gate. And so if either input is on, now the system is off. Further, you can take those promoters and very easily, just by swapping them, connect different inputs into that circuit. And this then produces different NOR gates. So if we have these promoters, we get a NOR gate. If we swap other promoters and so on, we always get a NOR gate. And importantly, you can predict the function of this NOR gate just by what the transfer functions of the inputs are. So you don't have to recharacterize the circuit every time that you connect two new inputs. It's behaving as you would expect from the additive combination of these different components. Just as kind of a demonstration of this modularity, we set up a simple system where we took colonies of cells on a plate, and each colony is carrying a NOR gate. And the NOR gate is processing signals that are being added to the media, and they're also communicating with their neighboring colonies in order to produce a computation. And so you can take NOR gates, and because NOR gates are what are known as Boolean complete, you can take any computational function, break it down to NOR gates, and in this case, connect up these communicating colonies such that they're able to produce that function. So for example, if you want to make an XOR function, what we do is we have 4 colonies. Each colony is carrying a different gate. They're communicating with each other using quorum signals, and then the inputs are being added to the plate. And XOR is exclusive OR is one of the more complex functions where if either of the inputs are off or the inputs are on, the system is off. If either of those inputs are on, the system is on. And in this system, you see the signal is going from left to right, and each cell is behaving properly, and then you get a very strong XOR gate at the end, where if either input is present, the system is turned on, system is completely off in either case. And so it's the same NOR gate where we're just rewiring the inputs in order to get these cells to communicate. The system's very modular, so by spotting the colonies in different spatial configurations and different compositions, you can make an XOR gate, equals gate. You can make all 16 different circuits just by connecting these different components together. None of these circuits had any additional genetic modification. It's literally just swapping these different components in and getting it to work. And so this is one of the first steps that we're going towards really having integrated circuitry going on in the cell. Okay, so one of the important concepts when you start to connect these circuits is to be able to make it such that the output of one circuit is in the proper dynamic range to connect to the next circuit. And this is related to a problem known as impedance matching in electrical engineering. And it's a very simple problem where if you have an input promoter that's driving an activator, if that input promoter is a little bit leaky, so that's always above the range that's required to produce enough activator to turn on the second promoter, the system is always on. And then if the first promoter turns on but never gets across the range that's required to trigger the next circuit, then the combined circuit is always off. So the trick when you're trying to connect 2 circuits in series is to somehow modify the transfer function of the first circuit so that it crosses the right dynamic range to trigger the second circuit. And this is what gives you a functional combination. One of the easiest things that you can modify in the circuit to change its dynamic range is the ribosome binding site, which is controlling the translation rate and controlling the amount of protein that's being produced. And so one way that you can look at it is if you have a weak ribosome binding site, then the circuit doesn't behave properly. If you have a strong ribosome binding site, the circuit also doesn't behave properly. And it's this optimum range where the activity or the strength of that binding site allows you to have a functional circuit. Previously, we had been using directed evolution as a method to go in and randomly mutate the ribosome binding site in order to identify one that would be able to functionally connect circuits. And we started out screening millions, and sometimes we got lucky with 4. The problem with this is it doesn't scale very well. So it's very hard to set up a directed evolution screen for every 2 circuits you're putting together if your objective is to be able to put 20 or 30 circuits together ultimately. It also gets very difficult to screen for the phenotypes that emerge from these more complex circuits like the edge detector and so on. And so what we decided to do is be more quantitative about it, where we developed a very simple thermodynamic model of all of the interactions that give a ribosome binding site its strength. And so if you want to calculate the energy of a ribosome binding site binding to a piece of mRNA, one thing you have to be concerned about is the folding of that mRNA, and you have to unfold it to make it accessible to the ribosome. And so we use a mRNA folding algorithm to calculate the free energy of that. The ribosome then binds to 2 locations, and so you have a binding strength associated with that, and then you have a stretching and compression energy based on how close or far apart these 2 binding sites are. And so once you have that thermodynamic function, you can then use a Monte Carlo search algorithm where you set a particular expression level that you want, and then it goes through a process of iterative mutation and calculating the energy in order to identify a ribosome binding site that's associated with that strength. And so you can see this here for a red fluorescent protein, where each one of these points is a synthetic ribosome binding site, where we dialed in different energies that we wanted and then measured the expression level that was produced. And so this gives us a way to dial in a particular strength that we want. And this becomes important when you're connecting circuits. And so we showed this with a AND gate that we had constructed, the same one as I showed previously, where we were attaching the PBAD promoter, which can be thought of as an arabinose sensor. And the problem was then identifying a ribosome binding site that would functionally connect that sensor to this circuit. And so what we did is, based on a previous characterization of the transfer function of the circuit and a previous characterization of the transfer function of the sensor, you can calculate the theoretical fitness function as a function of the free energy of that ribosome binding site. And then by picking different ribosome binding sites, we could then measure the activity of the AND gate as a function of the two inputs. And so you can see that we were able to recapitulate the fitness function. And so what this tells us is that if you have a transfer function for a sensor and you have the transfer function for a circuit, you can use a biophysical method like this in order to identify the correct ribosome binding site that's required to connect those two parts together. And so this will become more important as we start to automate the connection of circuits for very large systems. Okay. And so we've been putting all of this work together as part of a package where the idea, again, is only to build the controller bits, where each project has its own sensors that you're interested, each project has its own outputs that you're interested in, and we like to automate the design of the circuitry that connects those sensors to those circuits. I'm actually going to skip that and just talk about the last output, which will be important for the second part of the talk, where we have to somehow connect the circuits to the systems that they're controlling. And so in order to do this, we have what we call wires, which are just activators of some sort. Because what we do is have a controller plasmid that's on a low copy origin, and then we have all of the structural genes that we're trying to control on a compatible second plasmid. And so we need some way to link the activity of the circuits to those systems that it's controlling. And so in order to do this, we've been building orthogonal T7 variants, where one of the problems with the T7 polymerase is that when you express it along with a promoter, it can be very toxic. And so we went in and made a number of changes to the T7 gene. We added a degradation tag, made some active site mutants in order to slow it down, use a weak ribosome binding site, and so on. And then once we had that scaffold, what we wanted to do is to be able to go into the sequence databases to try to find orthogonal polymerases that we could use along with T7, but we didn't want to have to re-engineer each one of those polymerases in order to be non-toxic. So we identified a loop that interacts with the binding site in the promoter, and instead of taking the entire polymerase sequences out of the databases, we just took the sequences of that loop and just substitute that loop in and then screen for orthogonality. And these are usually very small numbers of changes, between 3 or 4 up to about 9. You then have to make a 6 base pair change to this T7 promoter, which then gives the binding site that's associated with that particular polymerase. And so the first system that we did was the T3 polymerase, where here you can see if you take the T7 promoter and you add the T7 polymerase, you get activity. T3 is inactive. If you just take the amino acids that are in that loop that bind to the promoter and swap them, now it activates the T3 promoter, but not the T7 promoter. We did this for some more exotic polymerases, such as K1F. And again, now that K1F promoter is turned on by its own polymerase, but not turned on by T7. and then the T7 promoter is not turned on by the K1F promoter. And you can really see this, I think, visually best here, where each of the columns is a promoter that has a 6 base pair substitution so that it will bind to a particular orthogonal polymerase. And then each one of these polymerases is based on the modified T7 backbone into which we've just put a few amino acids from each of the orthogonal pairs. And you can see when the output is red fluorescent protein, only the diagonal lights up, and that means that only those polymerases are interacting with the cognate promoter. Okay, so in the last 10 minutes, I'm going to describe how we've been applying all of this into rebuilding the nitrogen fixation gene cluster from the ground up. So nitrogen fixation has been one of the oldest problems in biotechnology. It's a major sink for natural gas and one of the bigger problems in terms of making agriculture more carbon neutral. And what you really want to be able to do is optimize the system so that it can be transferred between hosts ultimately, and also to get rid of some of the native regulation that do things like repress the system in the presence of ammonia. So the bacteria Klebsiella has a nitrogen fixation gene cluster. So it's 25,000 base pairs of DNA, about 20 genes. And it has all of the genes that are necessary for the scaffold of the nitrogenase, as well as the pathway that builds the FeMoco, which makes the nitrogenase active. And this is one of the more complex iron-sulfur clusters in biology. And then it has additional genes that deliver electrons to the nitrogenase, which are important for its function. And so this is the overall blueprint of the refactored nitrogen fixation gene cluster, where we have sensors that are responding to oxygen, energy levels, and ammonia, which are then integrated by a series of circuits. They then control different operons that are involved in the actual process of fixing nitrogen. And each one of these systems is split into a controller and a structural plasmid. So I won't go into this into too much detail, just to give you an idea of what the numbers are that I'm showing. The nitrogenase assay is very low throughput. There are no screening assays that have been developed. This is the rig that we have. You can do about 10 samples at a time, and it's based on acetylene to ethylene reduction. And everything's being reported as a % of wild-type activity in the system. And the low throughput nature of these experiments is going to come up as being more important. So one of the first things that we do when we're trying to rebuild the gene clusters, we do something we call a robustness analysis, where you take either individual genes or sets of genes out of the cluster, put it under inducible control, and then by adding inducer, you can see how the system responds from the over or the under expression of that system. So for example, if we knock out the HDKTY operon and then only include HDK on the inducible system, there's a maximum that's well below wild type activity, and then it tails off. NIFY had been shown previously to be not essential for function. But what we noticed is when you add it back, then you get a much more robust system, and it's able to recover wild-type activity. So while NIFY is not essential, it is required for making the system robust. These are the profiles for the electron transport, which you can see are quite robust, except up to very high levels of expression. And then the FeMoco biosynthetic pathway range from being very narrow in activity up to very robust. And there's one protein NIFX where if you include it in any overexpressed form, it shuts off activity. And so that system actually makes it more fragile. And so you have to be careful of those. So once we have this, we have a basic understanding of what the levels are required for each of the proteins in this system. Okay, so then we went through a very tedious process that we spent the first really 3 years of this project just trying to re-engineer the first operon in this system. And this is the HDKTY. It's the main structural proteins. It's less than 5,000 base pairs. It only has 4 genes. Each of those genes is codon optimized, and so on. We started out making a whole slew of variants where we were trying to control the expression levels very carefully. We then started applying some of the tools that were being developed at the same time to try to step through expression levels. And this is the type of thing that we would do, where you would go in and try different ribosome binding sites for one protein, move that up in higher activity, then do the second one and the third one. And you can see where you have to make a lot of different variants in order to even show some very basic activity. And so this sort of summarizes all of this data, where you start out with a system that you receive from the DNA synthesis facility. It has almost no activity. You then build it up. It starts to look like the stock market, whereas you add the next gene in, you drop in activity, build that up, drop in activity, and so on. And so after 3 years, many different variants, and on 1/5 of this cluster, we were at about 20% of wild type activity. And this is what really went back and started to feed into the concept that we really need to do this faster and more efficiently. And so to do this, we turned to a algorithm that had been developed in the 1960s for numerical optimization, back when it was expensive to do a function call for particular operations, like a sign operation. And so the whole idea in coming up with the optimization algorithm was to reduce the number of steps that you had to take. And in our system, our function calls are these nitrogen fixation assays, which are very slow. And so we have to reduce the number of function calls. And so we adapted this algorithm, which is known as Nelder-Mead. It's part of a simplex class of algorithms. So if you have 2 parameters that you're trying to optimize, you start out with several points, and then it looks like an amoeba, where based on the activity of those points, it then tells you where the next point ought to be. You reevaluate the activity at that point, it tells you where the next point ought to be, and so on until you get to a local optimum. And so experimentally, our parameters are expression levels of individual proteins. And so we can take data where if we have this particular expression level, and that's our best so far, we then take a different level expression, which is our worst, and our next best. And then the algorithm tells us where the next experiment ought to be. And then because we have these biophysical methods or lookup tables, we can then pick a genetic part that has that particular level of expression. And then that gets run and fed back into the algorithm, and it tells you where the next step should be, and so on. And so this is for 2 parameters, but when you have a higher parameter space, because you're putting together a 12-gene operon, it's able to handle that complexity well. And so here's an example where we were choosing promoters, T7 promoters, to control the EN proteins out of nitrogen fixation. We got back very confusing expression data where one of these points at a particular expression level of these 2 proteins gave us no activity. This one gave us 19% activity, and this gave us 32% activity. So that gets fed into the algorithm, which then tells you where the next point ought to be. It told us to go into this clear circle, but we didn't have a part to put us in that circle. So we picked the closest part that we had, which had a higher activity. That then feeds into the next iteration, and that's where we are now. And so that's the basic idea of how this works, where it takes the data that you have, and then it tells you what the next step ought to be to get the maximum amount of information for getting to an optimum. So with this and with other tools that we had been developing at the time, we burned through the remainder of the pathway relatively quickly. So HDKY is currently at about 20%. And what this is showing is that for each gene or set of genes in the full cluster, the way to read this graph is the bottom line is where we started with the first construct that we got from DNA 2.0. this tells us where we were at a previous talk, and then this tells us kind of where we are now. So you can kind of see the steps that had to go. And so with HDKY, they sent us a non-functional operon, and then we had to optimize it. We're up to about 20%. F, we're over 100%. J, over 100%. EN is getting up there. And these are the newest ones that we just got at the end of last year. BQ is up to 20%. And then this last operon took us 6 months to get from 0. And this is actually a bit of an interesting story where the last gene that we had problems with here was NIF-S. And as it turned out, this went back to an incorrect sequence in the NCBI database, where when we took this sequence for the gene cluster right out of the database, there were 2 nucleotide mutations in that gene that were non-synonymous, produced amino acid changes, and that was something like 1,000-fold less active than the wild type one. And so we're really kind of hitting our heads as to what was wrong with this one last gene. It turned out the sequence information that we had received was wrong, and that's going to come up more, I think, in these systems. So for this one, what we've done is we've knocked out each individual component or set of components, and then we've expressed the refactored version on a plasmid. And each one of these was optimized independently using IPTG. So if I had written the IPTG concentrations at the top of each of these bars, they would all be different. So you can't just all combine these together then into a single system. So in order to do that, we started using all of these components that we've been building, where instead of using different IPTG levels, we found a T7 promoter that we had made that would achieve the same level of expression. And so here are each of the components, where in blue we have the optimized level of activity for a particular IPTG concentration. And then in green, we have a single IPTG concentration where we've optimized expression by choosing different T7 promoters. And overall, we're a little bit under, but we were basically able to recover the activity back. And then once we had that, it became much easier then to combine these systems into circuits, into the full system. So this is actually where we are now, where we've completed the refactoring of half of this operon, or about 12,000 base pairs, where we have a completely single synthetic operon that has all of these components. All of the genes have been codon randomized, all of the ribosome binding sites are new, and so on. There's a second controller plasmid, and using this system, we're at about 10% of wild type activity. So we're recovering the activity slowly, but it's a completely different system, completely different DNA, and so on. And this is where we stand. And actually, there was a joke. We'll see how funny you find this. But one of my favorite jokes, we have this, at UCSF, we have these faculty lunches. And Ken Dill, who does protein folding, gave his faculty lunch. And he gave this kind of brilliant talk about these very intricate folding algorithms for proteins. And he had applied it, and he had folded half of protein G. And one of the faculty members asked, what would you do if you had double the computational power? And a guy turned around to me and kind of whispered, fold the other half of protein G. And so that's our joke for here. What would you do if the synthesis cost came down by twofold? We'd do the other half of, we'd factor the other half of nitrogen fixation. All right. So that's where we are with that project. And all of the individual components for the second half have been built. It's just a matter of combining them together, which is what we're currently doing in order to build out the complete system. And so with that, I'll just conclude by kind of showing where we are with DNA synthesis, where as I've been a faculty member at UCSF, the synthesis capacity has been growing very fast. This year, the first genomes have been synthesized. And our design capacity has been growing pretty fast, where we started out really only being able to design individual genetic circuits. We started getting better in putting those circuits together. And I think we've reached a rich point here where there are a lot of interesting functions in bacteria where you can now use synthesis and sort of these new concepts of design in order to go in and rebuild those functions where Ultimately 10 years from now, if you're the genome designer, you can imagine even taking all of these different refactor systems and starting to construct them in order to create your ultimate organism. Or I want to secrete protein, so I'll pop this piece of DNA in. I want to fix nitrogen, so I'll pop this piece of DNA in, and so on. So with that, I'll thank the people responsible. Karsten Tem is a bioengineering student here at Berkeley. He's been doing all of the work with T7 polymerase as well as nitrogen fixation. Jeff Tabor did a lot of our light sensor work and edge detection along with Ansome. Howard Salas built the ribosome binding site calculator, which we've been using a lot. And Alvin has developed the NOR gates and has been looking at how we can link them together in order to create more complex integrated circuits. So with that, I'll take any questions. I just need my water.
Are there any questions?
Hi, I was wondering, is there a fear of stocking local maximum and using the optimization algorithm?
Yeah, and actually you, sorry, so the question is, Nelder-Mead is certainly an optimization algorithm that only finds the local maximum. It does better than a steepest descent or similar algorithm, but it's still very much trapped. The problem is that if we got very ambitious with the number of experiments we could run, it would be maybe 100, right? That would be just like Carson would blow his mind and take a summer and do that, right? And so that is orders of magnitude below, often what the comparisons are for these different algorithms, right? And so actually we want to get as fast as possible to any maximum with as few of steps. And so we've actually gone in and we've been building a theoretical model that says of these different types of techniques, if I only have 10 steps, which one is going to get me the furthest with those 10 steps? And so it's a little bit of a different problem than if you're actually doing numerical optimization. It's closer to sort of design of experiments type thing, which is another whole field where you're trying to design the next set of experiments based on the information that you have and so on. So local minimum are not a huge problem. Yeah?
So I was wondering to compare this to like electronic devices. So
Yeah.
I don't know this much, that much about it in principle, but the difference between on and off is normally orders of magnitude for electronic devices in circuits and electronic devices. Is that possible? It looks like in some of your examples, maybe a factor of 5 or 10.
Yeah.
Is that do you have to have the parts better before you can really put them together in a big string?
Yeah, so sometimes we certainly do. So we, for like the oxygen sensors, the first sensors we built were kind of twofold, and that wasn't going to be nearly enough to start connecting a circuitry. So you start to fiddle with those sensors to increase the dynamic range. Once you get up to about tenfold, that's about what you need to reliably connect circuits. This is something that we're just starting to even have an idea of what we want. And the real problem is not the full change, but the distribution within a population, where if you only have a twofold change and you look at cytometry distribution, there's a large overlap between the on and off states. And what that can lead to are errors where the off population doesn't trigger the next circuit in series, and then this propagates, and if all of your circuits have that type of range, every layer in your circuit propagates that error, gets bigger and bigger, and then the ultimate calculation fails. And so the main effect of getting higher dynamic ranges is pulling those populations apart where everybody is going through the calculation identically, if that helps.
One more question?
Yeah, so I mean, this I'm sorry, go ahead.
No, go ahead.
I mean, this might seem kind of mundane to what you guys have already done, but I mean, have you thought about using these techniques to optimize recombinant protein expression? Because that seems like
Yeah.
would be awesome. I would love to be able to do that.
Yeah, so one of the neat things about Nelder-Mead is that it has, and the way we're applying it, is it has no underlying understanding of what the biochemical pathway is. There's no model that it's using in order to try to figure out where the next point ought to be. To it, it's just a vector of numbers, and then in our case, a efficiency, a percent efficiency, but it could be milligrams per liter protein. It's just another number. So you have a vector and a number, you have a set of them, and it just tells you what that new set of numbers ought to be. So what that allows you to do is to combine molecular biology optimization with all of the other traditional optimizations. So one of those numbers could be temperature. One of those numbers could be glucose feed rate, and different, more traditional pH, that type of thing. And it would optimize those as well as all of the numbers associated with the molecular biology. And so one of the things that we're doing is working with companies that are interested in that kind of combined optimization of the genetic system as well as kind of the traditional fermentation parameters.
If you have any more questions, feel free to ask Chris. I know it's a little after 5, but let's just join me in thanking Chris again.